Well, good afternoon, everybody, and welcome to my talk today. My name is Emily Mitzel, and I'm with Nelson Laboratories. I'm a consulting manager at the company, and I've been with the company about 13 years, specializing in reusable medical devices. And I'm talking to you today on some very key issues, very hot topics that are in the news and that are very important for manufacturers and also for healthcare facilities. So today we're going to be talking about some news articles that just came out in the past few weeks. We are also going to go into what needs to be done for all the validations. Then we'll go into more information regarding cleaning, disinfection, and sterilization validations for reusable medical devices. And then I'll be touching on some human factors information as well. A USA Today news article came out a few weeks ago, and it specified that more than 8 healthcare facilities have had outbreaks of hospital-acquired infection. This specific article blames the automatic endoscope reprocessors and stated that the instructions were followed. But this is, still under debate with this specific article. The US FDA has been working with medical device manufacturers to get acceptable validation results for these types of devices. An article from CNN a few weeks ago as well stated that 7 hospital patients at Ronald Reagan UCLA Medical Center were infected with carbapenem-resistant Enterobacteriaceae, or otherwise known as CRE, which is a resistant bacteria. The device that caused the HAI did not have specific FDA clearance, but it is a device that was improved but did not have specific reprocessing validations for this type of device. According to the British government, drug-resistant bacteria could kill 10 million people a year and cost up to $100 trillion a year by 2050. And the rampant global spread is going to be hard to contain. Even the president has gotten involved in the resistant bacteria issues. He has implemented a 5-year national action plan at a cost of $1.2 billion. New rapid tests to detect emerging resistant bacteria will be developed, and research for new antibiotics and vaccines will be accelerated. The most recent and important guidance for reprocessing for medical device manufacturers is the new guidance from the FDA. This is from a draft version from 2011, and it is their new version that just came out just a couple weeks ago. The major differences are the Appendix E, which specifies devices in which a 510K should contain data to validate reprocessing instructions, as well as the fact that 2 quantitative markers should be utilized for the cleaning validation and definitions of controls that are utilized in the validations. But this is a very key document for medical device manufacturers. And the FDA has been utilizing the information in this document since before 2011. So just a little bit about definitions. A hospital-acquired infection, or more broadly, a healthcare-acquired infection, is an infection that has been developed in a hospital. So this is an infection that a patient does not have prior to entering the hospital and has when they leave the hospital. These types of infections are the most frequent adverse event in healthcare facilities, and hundreds of millions of patients are affected by healthcare-associated infections worldwide each year. About 7% in developed and 10% in developing countries will acquire at least one healthcare-acquired infection. There's a lot that's being done to limit and decrease the amount of healthcare-acquired infections in healthcare facilities. There are a lot of standards that are and have been developed, and a lot of meetings that we have been doing for the past few years. We had the AMI FDA Medical Device Reprocessing Summit in 2011, And then, like I said, the draft came out in 2011 as well. So that was a lot of information from medical device manufacturers. We then had TIR 30 come out as well in 2011, and that's specific instructions for validating reprocessing medical devices. And then the 2013 FDA roundtable discussion, which I co-chaired, and we hosted that to make sure that we can decide how clean is clean, which I'm sure you've heard a lot in the news and so on. We had a couple standards come out just last year that were either redone or new standards, and that's water quality for reprocessing medical devices and also human factors engineering for medical devices. It also, in the spring, I think it was just actually last week, we had the document Flexible and Semi-Rigid Endoscope Reprocessing in Healthcare Facilities come out as well. I think that was just, it was either last week or the week before that this document came out. So that is brand new as of just right now. The healthcare facilities have also been doing a lot to decrease and reduce healthcare-acquired infections as well. These include special drying cabinets for clean scopes, utilizing ethylene oxide sterilization equipment for sterilization. They've also been focusing a lot on proper training, ensuring that all instructions are followed and that they have the right disinfectant, equipment, etc. for all the reprocessing steps of their reprocessing of medical devices. Also, there was a CDC surveillance protocol that was issued just a few weeks ago as well, and I'll be getting into more in that in detail at the end of the slide presentation. So the one thing that's really, really important as you start the process of validation is the written justification for the cleaning, the contamination, the sterilization, and everything, and the acceptance criteria, and everything that goes along with a validation. We find most often that the FDA is not, when they ask questions to a medical device manufacturer, it's not because what they've done is incorrect, it's because the justification has not been complete, or it's not enough for the FDA to really determine that there was a thoughtful process going into the validation concepts. The next step are the validations, cleaning, disinfection, and sterilization of every reusable medical device. And then the summary report with results and acceptance criteria stating everything that is meeting the acceptance criteria. So the first thing to think about is cleanability. And so when you look at cleanability, you look at the device design, the instructions for use, the compatibility of the device with the detergents, processes, etc., and also the human factors involved. So we want to make sure that the cleaning instructions are easy to follow for the healthcare facility as it goes into the next steps of the process. According to ISO 17664, a validated method of cleaning shall be specified. And this includes both automatic and manual cleaning methods, because the healthcare facilities may only be able to do one or the other, or they may have a breakdown of equipment, and so both methods need to be validated. Along with that, we need to make sure that all of the description of accessories required, temperature, concentration of solutions, exposure times, these need to be exact and specified in the instructions for use. We also need to determine all the techniques for rinsing, which could include flushing, any type of specific apparatus that's utilized for those types of processes. And then the identification of the water quality. Can this just be tap water? Does it need to be a specially manufactured water, etc.? And then we also needed to determine the amount of chemical residuals. So we are making sure that we are doing the proper rinsing to remove all chemical materials from devices. The next thing to think about is the fact that the cleaning, sterilization, and disinfection validations will be the easiest to perform on brand new devices. So we want to make sure that the devices are in more of a used state going into validation. So in this aspect, the FDA is requiring devices to go through some simulated use procedures to get the devices to be in a used state prior to the validation. FDA's biggest keywords right now are worst case testing and clinical relevance. So we want to make sure that we are doing the worst case testing in all of the parts of the validation. This includes determining a clinically relevant test soil, contamination, and all of the cleaning that we're performing to make sure that the cleaning is the minimum times, temperatures, etc. Here's a list of all of the soil ingredients and why they are clinically relevant. I presented this information to FDA in 2011, and we have not been asked about our soil determination since then. We also have been getting information from the FDA on, from our clients on deficiencies that other manufacturers have had, and we are finding that they're even recommending the test soils we utilize in the validation. So it's great news for us because we've written a lot of information on the justification of why we choose the test soils that we choose and ensure that they are simulated use test soils. The next step is the contamination step. We need to ensure that we are doing a simulated use worst case contamination. So we are performing a contamination exactly how the device would be utilized in a surgical setting. So we need to determine if the device is going to be, for example, immersed into the chest cavity, if it gets handled by a physician's bloody gloves, etc., and determining the exact test soil to be utilized. Will it be more of a mucus solution? Will it be a blood solution? Will it be a combination of the two? And we need to ensure that it's the exact use. The next thing, we need to make sure that the time that the device is in the test soil would be the exact time that the surgical procedure would be performed. And then the wait time after would be the amount of time that it takes for the device to get from the surgical procedure into the central processing area to be cleaned. Sometimes that could be a few minutes to hours, and sometimes it can be days if it's over a weekend time period. There are a number of ways that we can contaminate medical devices to ensure that we are performing simulated use studies. We can inoculate a fluid pathway by injecting the solution down the lumen. We can also spray the device, handle the device with bloody gloves, and also soak the device into a test soil. So as I mentioned, the dwell time could be any time from a number of minutes to hours to days. And this is sometimes the problem that we're seeing in healthcare facilities when they get so busy. They're seeing medical device trays stacked up in a certain area. And that is, so then we have to ensure that the wait time is going to be when the device soil is totally dried onto the devices themselves prior to performing the cleaning steps. After the cleaning procedure is performed, next is the visual inspection. Prior to just probably a few years ago, visual inspection with the naked eye under normal lighting conditions was a normal visual inspection that was performed on medical devices. As of the arthroscopic shaver issue in 2009, it has become more evident that we need to make sure that we're testing the inside of devices to ensure that the inside of devices are also clean, because those obviously cannot be seen by the naked eye. And in that case, we're using a borescope or also using magnification to looking at certain parts of devices like, screws and so on to ensure all of the parts of devices are visually clean. Here you can see a picture of visual human tissue that is found in arthroscopic cannula during an infection outbreak investigation. So that's what we would see with a borescope. So that's a very important aspect of the cleaning validation. After the visual inspection is performed, the medical devices are then extracted and then tested for residuals. The residuals that are tested depends on the regulatory submissions that will be performed on these devices. In the United States, the FDA requires protein and hemoglobin testing. Those are the most popular tests to perform on medical devices. They require 2 quantitative tests to be utilized. The EU is now asking for bioload reduction of a spore-forming organism, which is mainly Geobacillus styrothermophilus, and also Enterococcus faecium. And they're requiring that for all submissions for the EU, specifically for the Bavarian government. So we've seen this become very popular in the last few years or months, and so that's what we recommend for EU testing. Other tests that may be performed would be cytotoxicity to determine if there's any residuals left on the devices, as well as endotoxin testing if the device is coming into contact with any cerebral spinal fluid or brain tissue. Here I've got all of the acceptance criteria outlined, and these specific acceptance criteria mainly come from TIR 30, the AAMI TIR 30. The bioload reduction is from TIR 30, but like I said, FDA does not necessarily need bioload reduction information at this point. And what the Bavarian government is requiring now is probably a 5-log reduction. That's what they're asking for. It's kind of hard to meet. And so they seem to be happy with a 3 to 4 in some circumstances, but they are specifying a 5-log reduction as well. ISO 17664 also specifies for disinfection that a validated method of disinfection shall be specified, and includes mainly a lot of the same items that are necessary for the cleaning validation. And that includes all accessories required for the disinfection test, or the disinfection process of the reprocessing. Also, the specific contact time, temperature, concentration of the disinfectant. We need to ensure that the water quality for the final rinse is of like a critical water supply, since we are expecting these devices to be specified for a disinfected after that stage of the reprocessing. And we also need to test to ensure that there is no cytotoxicity after the disinfection test, to ensure that all the disinfectant has been rinsed off the devices properly. The testing for disinfection is based on Spalding classification, and this specifies that all critical devices have to go through a high-level disinfection process, and the acceptance criteria for this test is a 6-log reduction of a Mycobacterium species. For semi-critical devices, these devices need to go through an intermediate level disinfection, and the acceptance criteria for this type of disinfection is a 3-log reduction of the Mycobacterium species and a 6-log reduction separately of 4 vegetative organisms. And these are Klebsiella pneumonia, Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa. For non-critical devices, this requires a low-level disinfection and requires a 6-log reduction of the 4 vegetative bacteria that I just mentioned. Now moving on to sterilization, the healthcare facility can perform many types of sterilization on reusable medical devices. The most popular method of sterilization is steam sterilization, and then the next two are probably ethylene oxide and vaporized hydrogen peroxide. ISO 17664 also specifies that a validated method of sterilization shall be performed. And we also need to include the specific accessories required for sterilization, the concentration of the sterilant. We need to specify the humidity, temperature, pressure, exposure time, and any specific post-sterilization. So if we perform ethylene oxide sterilization, we have to perform aeration along with that type of sterilization. The steps for sterilization validation include sterility assurance level validation with half-cycle parameters. Then we perform a dry time validation to ensure that the devices are fully dry. And then we perform temperature profiling on the devices as well to ensure that they get up to the correct temperature during the exposure phase. An overkill study is performed on all reusable medical devices, since we do not know the quantity and types of organisms that will occur at the healthcare facility, because this will always change due to the fact that there are different patients involved, as well as different environmental contaminants in the healthcare facility. Biological indicators are placed in the worst case or the most difficult to sterilize locations, and these include lumens, mated surfaces, and cannulas. When we do sterilization validations on a large tray of, say, for example, a hip implant, we may test up to 15 sites or even more in this whole tray to ensure that we are getting all the most difficult to sterilize locations in the whole entire tray, because we want to ensure that the whole entire tray is sterile coming out of the sterilizer. For the reprocessing study, we want to ensure that every part of the study has a 510K approval. So this includes all the sutures, all the biological indicators. The sterilizer specific cycles that we're utilizing need to have 510K instructions for all of those, as well as the packaging. We want to ensure that every type of packaging that is utilized has a 510K for the specific cycle that we are performing. FDA is also specifying that only standardized sterilizer parameters must be utilized for sterilization. We are not able to just set the sterilizer for any type of parameters. This just causes the healthcare facility a lot of trouble trying to perform specific sterilizer parameters for every type of set of medical devices out there. Here's an example of the set parameters that are utilized for steam sterilization. And on your left, I have the most performed U.S. cycles and the most performed EU cycles. A lot of our medical device manufacturers that are submitting for regulatory approval in both the U.S. and EU are performing a cycle that is kind of in the middle of both these two, and that is we're performing a validation for a 132-degree cycle for 1.5 minutes as a half cycle. Therefore, they only have to perform one validation cycle, and then the cost is in half because of that. So that's a very good way for a medical device manufacturer to save some money and perform the sterilization cycle for that medical device. One more thing that I wanted to talk about was end of life testing. FDA is now requiring manufacturers to determine what that looks like. Since the healthcare facility does not always count the number of cycles every single medical device goes through, medical device manufacturers need to have specific instructions that state when a medical device is at its end of use life. So different ways to tell when the device cannot be utilized further. This is then determined by putting the device through a number of cycles. The type of device and the reprocessing instructions determine how this is performed. A medical device may have to go through the simulated use contamination procedure, and it may have to go through the automated reprocessor as well as sterilization cycles. So we work with manufacturers to determine what that will look like, to determine what the simulated use cycles should entail and how to perform that to ensure that we can get to an end of life cycle for the devices. Just a little bit of information on human factors and why they're so important is that the sterile reprocessing personnel are at the forefront of medical devices to ensure that the reprocessing instructions are followed correctly. So we need to ensure that the instructions that we are validating are definitely going to clean devices properly, but not be so over the top of ridiculously long and laborious to ensure that we can make sure that all of the instructions can be followed. So when I consult with clients to determine what needs to be done, we need to ensure that worst case testing is performed, but we don't want to go to more of a ridiculous case testing, because we want to make sure that the instructions for use are easy enough to follow for all healthcare facilities. There is a lot of instructions that have been placed into standards, guidance documents, etc. And I've listed those here. There's just so much going on with ensuring that we are thinking about human factors The FDA has also asked medical device manufacturers to go into healthcare facilities and watch the healthcare facility personnel perform the cleaning instructions on the devices to ensure that they are easy to follow and able to be reprocessed in a good manner. The surveillance testing for duodenoscopes came out just a few weeks ago as well. The CDC is requiring surveillance that will be, they're not specifying the time frame, but they said approximately 4 weeks to annually, or a certain number of procedures a year. These include cultivating the scope after the reprocessing steps, and then setting that scope aside until the results have come through for the healthcare facility. They are looking at specific high and low concern organisms that are outlined in this document. And this is a process that is we're doing a lot of research on this at this point. We have been performing this type of surveillance testing for healthcare facilities and scope manufacturers for a number of years now, and the FDA and CDC are now coming up with surveillance protocols for this. We are working with them to determine what needs to be done to, we've already validated on our end, but validate it more for the entire industry to ensure that this can be performed in healthcare facilities and other facilities across the United States and even the world. There's the FDA conference that's being held next week at FDA. Specifically talking about this, we are going to give a presentation about how we perform this testing and the validations associated. So this is a very, very, as I've shown you in the news articles at the beginning of the presentation, this is a very hot topic, and it's very important to ensure that we can work with healthcare facilities and manufacturers to make sure that we continue to keep patients safe. I just want to finalize the conversation today by talking about FDA's 6 criteria to follow. And this has come out of their reprocessing guidance that they just submitted a few weeks ago. And it states that labeling should reflect the intended use of the device. Reprocessing instructions for reusable devices should advise users to thoroughly clean the device. Reprocessing instructions should indicate the appropriate microbiocidal process. Reprocessing instructions should be technically feasible and include devices and accessories that are legally marketed. Reprocessing instructions should be comprehensive, and they also should be understandable. So it's very, all this information it makes a lot of sense, but that's what they're requiring for all medical device manufacturers at this point. And then I've just included some references for your information. There's so much going on with all this testing and documentation and so on. So it's very important to know the references associated with validations with the cleaning, disinfection, and sterilization. Well, thank you very much.